Hello students, welcome to the lab review. We're going to be going over some questions that I will talk about during the lab practical, so you can go over kind of what I'm talking about. Really quick, the lab manual has some questions at the end of the part 1, so we can just kind of go over that. So just at the beginning, we have practice conversion problems. You might just want to go over those real quick just to make sure that you know how to do those. The length, you don't need to use a metric ruler or anything like that, so you shouldn't need to do that. Temperature, you should know what the freezing point of water is, what the boiling point of water is, so make sure you know those in both degrees Celsius and degrees Fahrenheit. So what is the freezing point of water in Celsius? That's 0. What is the boiling point of water in Celsius? That's 100. Freezing point of water in Fahrenheit is 32. Boiling point of water in Fahrenheit is 212. Okay, what type of thermometer is always used in biology lab? We always use the Celsius thermometer. Okay, make sure you know how to use a triple beam balance. Okay, and how to measure between grams and milligrams. All right. Know what the term meniscus is. All right, so that's the little hook or the little bubble at the bottom of the graduated cylinder. Know where to look at that. What's the smallest unit of matter? That's the atom. What is a molecule? That's two atoms put together, that's bonded, right? Know what an empirical formula is. That's both a number of type of atoms in a particular molecule, right? That's when it's given in a formula. Know what the different types of sugar are, right? Pentose, hexose, octose, you know. So pentose is 5, hexose is 6, octose is 8. Functional group, what functional group is NH2? That's an amino group. Know what the different functional groups are, okay? COOH. So COOH is a carboxyl group. Then you should know what a hydroxyl group is. Hydroxyl group is an OH, right? How can fructose be distinguished from glucose? Fructose has the aldehyde. Glucose is an aldehyde, and fructose is a ketone, right? So you should know where that CO bond is, that double CO bond. How can glycine be distinguished from alanine? Glycine has a CH R group, and alanine has a CH4 R group. Is water inorganic or organic? Water is inorganic, right? What reagent was used to test for proteins? It's the Buret's reagent. What reagent was used to test for starch? That was iodine. How do you know if a substance was positive for starch? Well, it turned a blue-black color, a dark blue color. How would you test for lipids? You would use a paper towel, right? And you would put it on the paper towel, and if it would turn clear, it would be a lipid. It would leave a clear stain, transparent stain. How do onions store their carbohydrates? You would see it as monosaccharides, and how do you know? You would use the Benedict's test, right? The Benedict's test would test positive. Make sure that you know the potato as well. This was in your report, if you did starch. What is the pH scale based on? The number of hydrogen atoms in your solution, right? The pH scale is based on the logarithmics of the hydrogen atoms in solution. So if the pH paper turns bright red, what does that indicate? It indicates that the solution is acidic. Onto microscopes. What is it meant by parfocal? That means that the same amount of focus is equal on all objective lenses. Okay, so if you have one focus on one objective lens, if you switch to the next objective lens, it's going to be the same focus. Can electron microscopes be used to living organisms? No. They must be desiccated. If an objective lens reads 10 times, what is the total magnification of this lens? That's 100 times. Right? Because your optical lens is 10 times as well. So 10 times 10 equals 100. What domain does organism Annabana belong to? It's prokaryotes, so it's bacteria, right? Prokaryote or bacteria. You can choose either one. Do Annabana have a nucleus then? No, they don't. Does amoeba have a nucleus? Yes. Amoeba does have a nucleus. What is the difference between prokaryotes and eukaryotes? So prokaryotes don't have a nucleus. Eukaryotes do have a nucleus. Prokaryotes don't have membrane-bound organelles. Eukaryotes do have membrane-bound organelles. Right? You could also say prokaryotes, most of them have cell walls. Eukaryotes, some of them have cell walls. How do smooth and rough endoplasmic reticulum differ? Rough endoplasmic reticulum have ribosomes. Smooth endoplasmic reticulum do not. You can also say the smooth make lipids, the rough make proteins. What type of cell has a nucleoid region? Prokaryotes have a nucleoid region. Eukaryotes do not. Name the pigment inside a chromoplast. These are chromoplasts, not chloroplasts. So chromoplasts have carotenoids or carotene. Name the pigment inside chloroplasts. These are chlorophyll. Name the substance inside amyloplast. These are starches. Is euglena heterotrophic or autotrophic? This one, you can say both, but it can be autotrophic or both. Is Annabana heterotrophic or autotrophic? This one's autotrophic because it does contain chlorophyll. In a flower petal, the pigment anthocyanin is contained in what structure? This is the vacuole, right? Remember when we saw that? It's contained inside the center of the cell. That's the vacuole. Are plants irregularly shaped or rectangular shaped? Plant cells. Plant cells are rectangular shaped, right? They're boxy structures. Is an oral groove characteristic of an animal, plant, or protist cell? Oral grooves are protists. You saw that in Paramecium. Is a cell wall characteristic of plants, animals, or both? Plants, right? We don't have cell walls. We're not structured like that. All right. What is the function of the nose piece on the light microscope? That's to hold the objective lenses. I think that's most of the questions that I will go over here. If you have any more questions, please let me know. Other than that, good luck on your test next week. There's no assignments for today for this lab. So good luck on next week. All right.